One.
Good morning, Professor. How you doing?
I'm fine, thanks. How are you?
I'm doing good. I'm doing good.
Good.
Why is it my sister's name?
I beg your pardon?
I don't know why it's under my sister's name right now. Hold on.
I saw you changed your name.
Yeah, that's my sister's name. I think she might have been using my laptop.
Okay.
Wait, Professor, I have a question. When's our
Yes,
last when's our like final test or final exam? Like, do you know what
I have. No, I can't. Off the top of my head, I can't remember. But I'll certainly alert you. Your next test is on Monday. Next Monday, right?
I'm looking at the schedule
23rd.
now. Today is November 16th, right? Okay.
So you have a test on the 23rd, and then your final whenever it is scheduled by the college.
Okay. Okay, no problem. Thank you.
Okay. Anybody else got any questions? All right, then let's move on. We've talked extensively about mitosis, meiosis, and etc. We talked about Mendelian genetics. Now we want to try and put together a story about the genetics and what we observe physically inside the cell. and this we term the chromosomal basis of inheritance. We realize that by far the majority of our genes are carried on our chromosomes, which reside in the nucleus. Not all of our genetic information. We have a small amount of genetic information, for example, inside the mitochondria. But the vast majority of the genetic information is contained in the DNA sequence of the nucleus. But that was not always clear. To us, it is. In fact, we can do very refined techniques to suggest this is the case. Have a look here. And this is a human karyotype, all the human chromosomes laid out. And what they've done is they have actually been able to stain one particular gene, these yellow dots here, there, 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 and there. This is one homologous pair of chromosomes. When we look at them, we realize that chromatid there is identical to that chromatid there. These two genes are identical. This chromatid here is identical to this one here. That gene is identical to that. But these genes are homologous to these. They are not necessarily identical. They code for the same character, but the trait may be different. If this was eye color, this could be brown, this could be blue, whatever. So this realization for us means that we can understand when we look, for example, at I just need to move everybody's pictures out the way for a moment. When we, this is not working. Anyway, hold on. There we go. When we look inside the cell and we realize the genes are indeed located on chromosomes, we can understand really easily, if we understand mitosis, how it is that genes get segregated. We understand, for example, using the previous karyotype that we saw there, let's just take one set of chromatids, say it's one of the homologous pair. It has a homolog, but during the course of meiosis 1, those chromosomes get entirely separated from one another. Then in meiosis 2, the individual chromatids get separated from one another. What we end up with is we start off with one cell. In meiosis, we end up with 4. And each of these chromatids, 1, 2, 3, 4, is distributed to each of the cells. Now you need to remember that in human beings, in female meiosis, only one of those cells survives. And it survives as this very large egg with all of the material that's needed to see it through its initial divisions. So we end up, there are 4 cells produced, but 3 of them are sacrificed. In the male, that's not the case. Each of those forms a sperm. And in our simple example here, each of the sperm would have a chromatid included in it. For us, this is obvious. For us, understanding the nature of meiosis, understanding the basic rules of Mendelian genetics, we can see how these individual genes get segregated off into the gametes and then get recombined in the various combinations. This was not always obvious. It wasn't always obvious to biologists. In fact, it was only in the 19th century, really, that the process of meiosis was followed under the light microscope. And at that time, in actual fact, for much of that time, no association was made between genetics and chromosomes. They had no idea what the chromosomes did. They had no idea why during cell division they condensed the way that they did and seemed to separate the way they did. It was only when they actually looked at the process of meiosis and realized that at the end of meiosis, each of the cells that results from meiosis contains half the number of chromosomes and half the amount of genetic material, that the association between chromosome behavior and the behavior of what Mendel had called hereditary units, which we now know as genes, it was only then that they began to put that together. These two, Sutton and Bovary, but there were numerous others, began to make that association. They began to look at genetics and say, we can explain it if we explain the behavior of chromosomes during meiosis, because that explains the segregation of the genes, their separation, and then they're coming together again when we cross-fertilize, for example. But it lacked really, it was mostly theoretical. They were looking down the microscope, seeing how chromosomes behave. They were breeding their pea plants or whatever they did, and they were putting the two ideas together, but they had no experimental proof that what they believed was actually true. And that was really provided by the work of this man here in the early 20th century, Thomas Hunt Morgan. And Morgan is really famous because he established one of the first really important laboratory animal models for biology. And that was that he began to use fruit flies as an animal model. And he began to examine their genetics. So this is a fruit fly is incredibly common. Its name is Drosophila melanogaster. And there are huge advantages to working with it because it's very easy to keep. You just provide it with food in the form of porridge in a tube, and they eat themselves fat and happy. Put them together, both sexes. They breed really quickly. They produce lots and lots of offspring for us to count. And Morgan began to accumulate particular mutations. He looked for particular phenotypes and bred them over and over and over again until he could predictably breed particular phenotypes.
Professor?
Yes.
How was he able to see their mutations if the fruit flies were so small?
Oh, you can see them easily under the microscope.
Oh, I see.
I'll show you in a minute. There are many of them, but we'll look at one in particular.
So were they analyzing their actual physical their physical appearance, or were they looking at their genetic appearance?
No.
They're like DNA, I mean.
No. He knew nothing about DNA.
Okay.
Nothing at all. But what he could see was that there were particular phenotypes of the flies, and he could trace those phenotypes just as Mendel had done with the pea plants, except he was now doing it in an animal model. And in addition, there was one advantage. Another advantage to using flies is they actually have the same sex determination system as we do. The males have an X and a Y chromosome. The females have two X chromosomes. This was very fortunate for him, as you'll see in a minute. The other thing
Professor?
Yeah.
Sorry to cut you off quickly. I was just about to ask. So Morgan is basically doing the same thing as Mendel did with the charts and stuff, but instead of him doing it with P, he's doing it with a dragonfly, a fruit fly.
With fruit flies, yes. And he, at the same time, is looking at their chromosomes. There's a big advantage as well because they only have 4 pairs of chromosomes. So he could identify the chromosomes, the homologs, and everything very, very easily. So first of all, he looked at the general population with all of their normal characteristics, but he saw that every now and again in the population, one would arise with some distinct difference. And he could actually breed them and predictably see when this difference would appear, and basically following Mendel's rules.
Oh, okay.
So he called the normal, he called the wild type, and then he referred to these other phenotypes as mutants. And the very first one he discovered was actually one of his most important. and this is a fly that has white eyes instead of red. So here's a red-eyed phenotype here. That's the normal. But every now and again, you get this white eye here.
Professor, would that be known as albino?
No.
No?
It's not albino. No. No. It's just that character. All the rest of the pigmentation is normal. It just doesn't have the red pigmentation in the eye. That's all. It's a very distinctive characteristic, very easy to see. So he could sort them out really, really quickly. He looked at the pattern of inheritance. And so for example, he mated males with white eyes with females with red eyes. And that F1 generation, they all had red eyes. So what does that tell you about the red character? It looks dominant, right? It looks like that's dominant because all the F1 generation had red eyes. Now he breeds the second generation. And please note on your notes, please put a line through that. Okay, that's wrong. Ignore that, end of that sentence. Because the F2 generation had 3:1 red to white eye ratio, 3:1, but only the one with the white eyes was male. He knew that the females have 2 X chromosomes and that males have 1 X and 1 Y. That had been observed under the microscope. That males, their chromosome karyotype looked different. They had one pair where they had an X chromosome and a Y chromosome. Females had 2 X. He reasoned that this character must be carried on the X chromosome. The white eye allele must be carried on the X chromosome. It looks dominant here, but it appears in the males because they only have 1 X chromosome, and that X chromosome is carrying the mutant allele. I'll show you now. So let's have a look, and you see here. Here's the parental generation. Females, red-eyed. Male, white-eyed. All the offspring have red eyes. Now he breeds the offspring against one another, back and forth, gets lots and lots and lots of them, and counts up the phenotypes. And this is what he has in the F2 generation. 50% female, 50% male. All the females are red-eyed, always. They are never white-eyed. Here in the males, 50% red-eyed, 50% white eye. He reasoned the only way to explain this is that these characters are being carried by the sex chromosomes, not by all the other chromosomes that we call the autosomes. This character is carried by the sex chromosomes. And here's how it works. Here's our female. Here she is. She's pure breeding, and she's got red eyes, W+, never mind why it's W. It just is. She's got the red eye character, W+. Here's the male. He's white-eyed. Morgan reasoned that this was a recessive allele, and it was carried on the X chromosome that the male has, because males have X chromosome and a Y chromosome. They're not carrying any of the genes. They don't carry any of the alleles on the Y chromosome. Their only source for the allele is X chromosome. This is a white-eyed male. It's a recessive allele. He must be carrying the recessive allele on the X chromosome. There it is there, little W. So let's work out a little mini Punnett square for the F1 generation. The female, the only eggs she can produce is W+. That's the only allele she has. She's homozygous. She can only produce eggs with W+. The male, on the other hand, there's 2 kinds of sperm he can produce. The one sperm carries the X chromosome. The other sperm carry the Y chromosome, 50%, 50%. Let's make these W plus, little w. There they are, W plus, little w. Here's the Y sperm, going to make male flies. But look, they inherit W plus from the female and the Y chromosome. These are all red. She's heterozygous, but none, it doesn't matter. This is dominant. Her phenotype is red eye. Here.
Wait, so Professor.
Yes.
Just to clarify on something, so only females in this case have W plus, which is just the X, right? And then the males have the X and Y, which means that they're the lowercase because they're not the dominant one. If that's
correct. This is the recessive. That's the nomenclature for the recessive. That's the red eye characters, little w.
Okay.
I mean, I'm sorry, for the white eye, it's little w. Okay, that's the white eye allele there, little w. All that they can produce is little w, sperm and Y, with no genes on it. Okay, so everybody here is going to be red-eyed, 50% male, 50% female, and all of them red-eyed. But now we're going to breed them against one another. What can these males, what kind of sperm can they produce? They can produce W plus and Y. But what can she produce? She's heterozygous. Her eggs, W plus, and look there. There's the mutant allele. She had it hidden here. We refer to her as being a carrier. And this is a very typical inheritance pattern for these sex-linked genes. This is a very typical feature that in the females, the allele is hidden because they have another X chromosome. And the X chromosome's got the dominant allele. So this recessive allele is hidden away. But now, look at her eggs. She can only produce W plus, red, and white. He's producing red and an X with none of the alleles on it. Let's fertilize them. Homozygous W plus, W plus, W plus. Heterozygous W plus, little w. There we are. Here, W plus, Y chromosome. It's as if he's heterozygous, right? There's nothing on here. He's only got the W plus. He's going to be red-eyed. But what about this guy here? He's got the little W there, a white eye, and there, a Y chromosome. He's got no other allele. He's only got the white-eyed allele. So he is white. He appears white eye. We got 1, 2, 3 red-eyed to 1 white-eyed. We've got 50% female, XX, XX, 50% male, XY, XY. And half of the males are going to be red-eyed, half of them are going to be white-eyed. This was really important finding because, first of all, it demonstrated conclusively that these chromosomes are actually determining all sorts of things. They are determining sex of the flies. That's number one. Remember that nobody had been able to conclusively associate chromosomes with inheritance up till this point. But Morgan is also showing that these chromosomes can carry alleles. And that especially in the male, if this is a recessive allele, which is carried on an X chromosome, then it gets expressed in the males because they don't have a matching X chromosome. They only have the Y chromosome, which is not carrying the homologous gene on it. So they only have one copy. Whatever copy they end up with on their X chromosome, that is what is expressed. In the females, they have the chance of being heterozygous and always having at least one copy, if not two, of the dominant allele present. So this is conclusive proof that genes are carried on the sex chromosomes. And it's also conclusive proof that these sex chromosomes are what determine the sex. And thirdly, that it establishes the strange pattern of inheritance where this recessive gene is present in females here, but has no impact on their phenotype, only to make its reappearance here in the F2 generation, because the males only have one copy. They only have a chance of achieving one copy, and it's either going to be the mutant or the wild type. Here, white, red. Okay, so use this diagram. I know that when I just tell you about it, it becomes confusing. Use this diagram and go through this carefully and realize right at the very start that as soon as you get an X chromosome, a Y chromosome there together with the X, it's that X that is going to determine the phenotype for these genes, not the Y. The Y does have some genes on, we'll talk about it in a minute, but the X chromosome carries these particular sex-linked genes. So this led to an explosion of interest in science about the nature of chromosomes and about how chromosomes actually carry information, the hereditary units that Mendel had talked about, what we now know of as the genes. And we and most other mammals, all other mammals that I know of, have two types of sex chromosome. We have this large X chromosome, and then in males there is a smaller, much reduced Y chromosome. People who inherit two copies of the X chromosome usually develop anatomy that we associate with female. I say usually because there are some strange exceptions, so it's not, sometimes there's some little differences that arise, but usually two X's determines female. Male properties are determined by the inheritance of the Y chromosome. And I will say this is not the only sex determination system. This XY system is common. It does occur in the flies, as we've seen, in all mammals. In other things like birds, reptiles, also, they have different systems. I'm not going to discuss them. If you're interested, you can have a look in your book. It does discuss them. So here are the chromosomes. This is the big X, just looks like a normal chromosome. And here's the Y. It has these two arms here, little short stubby arms, and then what usually looks like a little short stubby tail there. Carries very few genes. The genes it does carry, at least some of them, are very, very important. Others are not important.
Hey, so professor.
Yeah.
So in a guy, in like a male, the Y one is what we have as our genes, and the girls have the X.
Correct. Females have two X chromosomes. Males have X and a Y.
Oh,
and
wow.
there's a gene on here, which is the sex determination gene, which actually leads to the fetus developing the morphology of the male. The default position is female. That is all fetuses commence development, and if they don't have the influence of this chromosome for one reason or another, they will develop as female. But as soon as you have the sex determination gene here operating, and it gets the fetus to respond, to produce and respond to testosterone and that drives the development of the male morphology. So they still behave as homologous chromosomes, even though they don't carry homologous genes on them. They have areas on them which are actually homologous and allow them to perform correctly during mitosis, and then especially during meiosis, to segregate and do all the things they are supposed to do during meiosis. Now, there's this little short segment, this gene on the Y chromosome, which is called SRY, sex determining region on the Y chromosome. And that is what drives, if that is present, it drives the morphological development of the male anatomy. But the baseline anatomy is female. If for any reason at all, this gene is not operating properly, or if, for example, it happens that sometimes the fetus does not respond to the presence of testosterone, the fetus will develop as female, even though they have a Y chromosome. So there are instances, well-known instances, where people have X and Y, but for one reason or another, they develop as female because that is the default. We are all fundamentally female. The Y chromosome imposes the male morphology a little bit later on in fetal development. So any gene that's located on one of the sex chromosomes is called a sex-linked gene. And there are some genes on the Y chromosome, SRY, for example, which is a Y-linked gene, but by far the majority of sex-linked genes that we are interested in are X-linked. Now, on the Y chromosome, there are about 78 different genes, and they code for just a few different things, but very importantly for the sex determination. That's the most important responsibility of the Y chromosome. And the X chromosomes, they're completely normal chromosome. It's the normal size. It has about 1,100 genes, far, far more than the Y chromosome has. Now, I just told you that the Y chromosome actually, it has some very strange things about it. It has some strange genes on it, and this is one of them. I just put it here for amusement's sake. It has a gene for hairy earlobe. There it is there. This guy looks like he's trimmed it. But you will see this, keep your eyes open on the subway. You will periodically see this, not that common, but this is a Y-linked gene for hairy earlobe. Why? I don't know. Have no idea whatsoever, but there it is. So this, obviously, hairy earlobe, you're not going to get hairy earlobes in a female. You may have for entirely different reasons, but you're not going to get it from this gene. Why? Because this is a Y-linked gene. It's only on the Y chromosome. And this, if it's on the Y chromosome, again, you're going to express it because there's no matching dominant on the X chromosome that might cover the phenotype. Now, this pattern of X-linked genes, it results in a very, very specific pattern of inheritance, and which we see over and over again for recessive traits. And the reason for that is a female, in order to express that, the recessive, she has to have two copies. She has to have a copy, she has to be homozygous on the X chromosome to express the recessive. A male, on the other hand, only needs one copy of the recessive because he's only got one X chromosome. He doesn't have another X chromosome to provide a dominant. He's only got one X chromosome. If that X chromosome is carrying a recessive gene, he will express it. And for this reason, these recessive sex-linked disorders, X-linked recessive disorders, are much, much, much more common in males than in females. They do occur in females. I'll show you how in a minute. They do rarely occur in females. But because females have a much higher chance of having at least one copy of a functional X chromosome, they very seldom express the recessive. So there are a few conditions which are very famously associated as sex-linked genes. And here is one of them. And this is a relatively harmless one. This is the recessive gene for color blindness. I think red-green color blindness. And here are three different scenarios for you to look at, possible patterns of inheritance. So our first one, here's a female, and she's homozygous, and she is completely normal vision, big N, big N. Here's a male. This he is expressing, you can, so the square is colored in to show you the dark color shows he is actually colorblind. So he has only one X chromosome. Here it is here, and it's carrying that recessive allele for colored blindness. He doesn't have another X chromosome. He only has a Y chromosome. So that has to be expressed. That's why his phenotype, he is colorblind. Now let's have fertilization. His sperm, what sperm can he produce? He can only produce XN there and Y. XN, the mutant, the color blindness, and Y. The female is homozygous and she has normal vision. She just produces XN, XN. Let's fertilize, and what you get, big N, little n, heterozygote. Big N, little n, heterozygote. Both of these are female. Why? Because they have two X chromosomes. There and there, there and there. Here, however, we've got two males, Y males, because they inherited a Y chromosome. They only have one copy of the X chromosome, and there it is there. They got it from the mother, XN, XN. Bingo. Is anybody colorblind? No. No one's colorblind. Why? Well, look at the two males. They both have the normal allele there that they inherited from the mother.
Wait, so I have a question.
Yes, go ahead.
Do you see how on the diagram you're looking at right now, on the left side, there's two boxes that aren't fully shaded, but they're like shaded a tiny bit?
Yes, I'll tell you right now.
Oh, okay.
I'm going to get there right now. Yeah. Okay. Yes. So these are normal, but look at these females. They are heterozygous. Big N, little n, big N, little n. They are carrying the recessive, and that's why this is lightly shaded. These females are what we refer to as being carriers. There's no expression in their phenotype. Their vision is completely normal. Why? Because they have this allele present. But they are carrying the recessive. And let's see what happens now when we take females like this, and we start to see how their progeny might turn out. Here we go, here they are. Big N, little n. She's lightly shaded because she's a carrier. Now we're going to breed with a completely normal vision male there. Now look what happens. What sperm can he produce? Y, normal allele, normal vision. But what does she produce? She produces eggs that are either normal, big N, or the mutant recessive allele, X, little n. Now let's take her eggs. Those are her two kinds of eggs. Here are his two kinds of sperm. Let's fertilize them. Heterozygous, big N, little n. This one here. I'm sorry. There's a mistake here. There's a mistake. I just spotted it. That should not be little n. That should be big N. Make a note. X, big N, big N. That should be big N. She should be homozygous, normal. But look here, heterozygous. This is female again, and she's a carrier. Look at the males. He inherits the normal phenotype from the mother and a Y chromosome, normal vision. But look here. We've got Y chromosome and the recessive allele. This child here is going to be colorblind. They've inherited the recessive allele from the carrier mother, and they've inherited a Y chromosome from the father. So this must be expressed. This is typical. This is a typical pattern of inheritance of these X-linked. They remain, the females are carriers. The phenotype remains hidden until it appears in males, usually in the second generation. And we're going to see, I'll show you a real family tree in a while where this is true. Yeah, go ahead.
Sorry for the question.
No, why be sorry? No, go ahead.
So with this whole diagram, the gene that has the color blindness is the X and then the small n, right?
X
and then
small
it's
n,
on the,
that one
and
there.
it's on the Y chromosome.
No, it's on the X.
Oh, okay.
The Y doesn't carry any.
Oh, okay.
For this gene, you can just assume there's not, the Y is not carrying any genes that we are interested in this example.
Okay, so how do you know 100% if they're colorblind or not? I'm just kind of confused on that.
You can test. You can very easily test. You can test to see whether they can distinguish between red and green, for example. People who are fully red-green colorblind can't distinguish between red and green because they don't have the cells in their retina that will do it.
Okay.
Okay, so this is very typical. This is a very typical pattern of inheritance when you have this recessive gene sitting on an X chromosome, female carriers, and then it pops its head up every now and again in the males because they only have one. Let's take another example. Here's, again, same thing, heterozygous, big N, little n female, 2 X's. Now we're going to fertilize with a male who is colorblind, same as this here. Okay, here he is here. There's the recessive allele there sitting on his X chromosome. He's only got a Y chromosome. What can he produce? Big Y, little n. What has she got? She's got big X, big N, little n, big N, little n eggs. Cross them. Big N, little n, X, X, female, carrier. X, Y, big N, male, normal vision. Here, little n, Y, it's got X, Y, little n, colorblind. But look here. We have little n, X, X. In all of these, we end up with one female who is colorblind. And the reason that she's colorblind is we have a recessive here in the male, and we have a hidden recessive here. And you run a chance of having a homozygous recessive. She's homozygous for her sex chromosomes, XX, homozygous for the mutant, N, little n, little n. And she is that rarity. She is a female who's expressing this mutant allele. This would be, you can substitute any number of disorders for the color blindness. This applies to many, many disorders which are carried in a similar way. Whenever you have a recessive carried on an X chromosome, you see these similar patterns of inheritance. And here's a very famous one. This is the royal family. This is the royal family in starting in Britain. This is Queen Victoria. There is her husband, Prince Albert. And none of his family ever showed this condition of hemophilia. Yeah, go ahead.
Is this the story about that family where the young boy had that disease where he couldn't get bruised? Yeah, because he was bleeding easily, and then they all got murdered.
Exactly. Yes. Okay. Yes. Yeah, because his great-grandmother was Queen Victoria, and here she is here. Her husband Albert, his family had no hemophilia in it at all. This condition of hemophilia is a defect in the clotting mechanisms of the blood, and it is inherited as an X-linked recessive gene. So it is believed in this family to have originated here. There was no trace of it before in her parentage. So it arose spontaneously here in Queen Victoria. And if you look at her children, one of the sons, Leopold, actually had hemophilia, and the rest did not. Here are the females. Alice, I don't know who that was. Oh, this was also Victoria, and Beatrice. Now, Alice and Beatrice are of interest because when you look at their children, this is what you see. Here's a male, one of the princes, son of Beatrice. There's another one there, both of them hemophiliac. She has a daughter, but the daughter doesn't express it. Why doesn't the daughter express it? She doesn't express it because she's got 2 copies of the X chromosome and it's only being carried on 1 of the X chromosomes. But look at her children. She's got 2 sons, both of whom are hemophiliac. She's got 2 daughters with, we don't know. But she's got 2 sons that definitely express. It's the typical pattern of inheritance. Here it is in Victoria, expressed in her male children, not expressed in her female children, but the female children are carriers. We know that they are carriers because it appears in their children. And we know that this one is a carrier because it appears in her male children as well. Same here. You can see how it pops up all over the place. Here's Alice. She's a carrier. We know she's a carrier because one of her sons is hemophiliac. Her daughters, here they are. They show none of the phenotype, but we know that they are carriers because their own male children, some of them, express. Look here and you'll see this one does not. But that's right. We expect that some of them will not. They have a chance, their males have a chance of inheriting 1 of 2 chromosomes from the X chromosomes from the female. They may just be lucky enough to inherit the normal X chromosome. These guys here did not. They inherited the X chromosome with the recessive allele. And just to finish the story, this here is the current royal family line here. This here is the current Queen's father, George VI. That's just his father, George V. That's his father, Edward. None of them show the hemophiliac allele at all. None of them. So they are lucky because they've escaped it. The rest of the royal family in Europe was not so lucky because at this time, this is the turn of the century, 19th, 20th century, the royal families of Europe were actually extremely interbred. And there was many instances of cousins marrying cousins and everything. And this gene for hemophilia spread widely through the royal families of Europe, including, as just heard, the Tsarevich of Russia. and he inherited it from his mother, who was related to Queen Victoria. She was a granddaughter of Queen Victoria.
Wait, Professor?
Yeah.
So is it possible that Albert and Victoria could have been a carrier also, because they could have went back in history and they could have had it, and just they didn't have to have been a carrier?
That has been done. Geneticists have looked at the families on both sides, and there's no appearance of it at all.
Oh, okay.
In either of the families, which is very interesting, and it suggests when we trace all of this back, we end up with her, and then it's a dead end, and that suggests that it originated. We know that it can arise spontaneously. This mutation does arise spontaneously, and it seems to have risen here in Victoria, not before. There are other color blindness we've talked about. There's another disorder called Duchenne's muscular dystrophy, also inherited in the same way. There are many, many other X-linked things which show the same pattern, whether they are harmless or not. In the case of hemophilia, it can be an extremely devastating inheritance. Not so much modern times, because we are able to treat it. This time, it was a death sentence. Many, many, many of these people, rich and famous and powerful as they were, many of those men died extremely young as a result of their hemophilia. Okay, here's another interesting little quirk about the X chromosome, and it's this. In females, there are 2 X chromosomes. But in actual fact, only one of them is ever operative in one cell at one time. So what happens is, when the egg is fertilized, female, and it's female determined because it's got 2 X chromosomes in every cell. After a certain number of divisions of the zygote, there's a process whereby one of the X chromosomes is silenced, and it's random. It's inactivated. It's called random X inactivation, and it's a random process. So you may have, I don't know exactly what stage it is. Let's say we have 32 cells, or then maybe 16 of them are the one X chromosome, and then the other 16 are the other X chromosome. And the one that is inactivated, it shrivels up, and it condenses into, you can actually see it on a light microscope. If you look carefully at a woman's, the nucleus of a female cell, you can sometimes see it. It makes a little dense staining blob in the nucleus, and that's called a Barr body. And it still does participate in mitosis and everything, but all of its genes are silenced. And this leads to a very interesting situation, because if a woman is heterozygous, if her one X chromosome carries one allele and the other X chromosome carries an entirely different allele, then we have both cells expressing. Now, sometimes that is very useful, and I'll show you exactly why. Have a look here. This is why these carrier females here appear normal. Just imagine, I know that we did this pedigree as if it was for vision, for color blindness, but this could as easily be for hemophilia or anything else. Let's just say it's for hemophilia. Here is a carrier female. She has 2 X chromosomes. She has one normal one there, and then she has another one there, whose allele is for hemophilia. What is in fact happening is in these chromosomes are failing to produce a clotting factor. They're simply not producing the protein that is needed. but she has another normal chromosome, and that is enough. The functioning of this chromosome is enough to provide this female with all the clotting factor that she needs. So she doesn't even know usually that she has one X chromosome, which is not functional. But on the face of it, approximately half the cells of her body are producing normal clotting factors. Approximately half will not be producing the clotting factor because they've activated that X chromosome. The other half have activated that X chromosome. So this is a very interesting situation, and we can see it very clearly. And the easiest is in these lovely tortoiseshell cats. And just think of it the way I described it to you. Let's say we got a ball of cells in the zygote, maybe 32 cells or whatever, and they undergo random X inactivation. Now, each of those 32 cells then goes on and on and on and on and on. They go on and on and on, dividing. All of their progeny inherit that same X as is activated. So what it leads to very often are these patches of cells which share a characteristic. And this is the classic example, tortoiseshell cats. Here's an allele for orange fur on the X chromosome. Here's an allele for black fur on the other X chromosome. Now they go through some cell divisions in the zygote and then X inactivation. Look at this one. This orange one is inactivated. Only the black one is activated. This one here, some other cells, the orange is activated, the black is inactivated. Look at everything that arises from them, and all the cells that arise from here produce orange fur, and all the cells that arise from here produce dark fur there. And that explains this mottled color. Could you get this in a male? And the answer is no, you can't get it in a male, because the male only has one X chromosome operating at a time. It's either going to be black or he's going to be ginger, orange, right? He only has the chance of inheriting one of these that is going to be functional. So that's just, okay. So now let's also think for a bit about some of the other implications of the chromosome theory. One of them is, in the normal functioning organism, us, for example, we are 2N, we are diploid. We have 46 chromosomes, we have 23 pairs of chromosomes. That is our normal complement, our normal ploidy, we say. Our normal functioning ploidy is that we are diploid. Our gametes are haploid. But in some organisms, it is actually possible to have more than diploid. You can have triploidy, which is 3N, 3 sets of chromosomes, and you can also have tetraploidy, 4N. Some higher than that have been recorded, but they're all recorded in plants, not in animals. These polyploidies are not well tolerated in animals at all.
Professor?
Yes, go ahead.
These count as mutations, or this is normal?
No, they're not mutations, no. These in plants, in actual fact, it's completely normal. I'll give you some, for example, bananas are triploid. And there are many other ploidies in plants. The only thing is these uneven ones, triploidy, 3N, 4N is fine. It's easy to undergo meiosis. 3N, it's difficult to undergo meiosis, and these triploid plants often don't reproduce sexually. So cultivated bananas, which are triploid, they are cultivated asexually. They take cuttings from them, etc., to grow them. In plants, these polyploidies often produce very big cells and very big plants. So these polyploidies are often encouraged in horticultural plants so that we get big plants, big fruit, etc. But in animals, it doesn't work. Polyploidies are not well tolerated in animals at all. Any sort of changes in number that are associated, especially with fertilization, with production of gametes during meiosis, and with production of the zygote, any changes in chromosome number is referred to as aneuploidy. And the commonest aneuploidies are actually differences in the number of a particular homologous pair. Either you end up with 1 instead of 2, or you end up with 3 instead of 2. Those are typical aneuploidies. But these aneuploidies have particular phenotypes. Now, I'll just tell you that in human beings, these aneuploidies are very, very poorly tolerated. There are very few of these aneuploidies in humans that are where the fetus will survive an aneuploidy. And these mistaken pairings and everything of the chromosomes are a common cause of spontaneous abortion, of loss of a fetus due to miscarriage. There's one exception to this, and that is the sex chromosomes. We are very, very tolerant of aneuploidies in the sex chromosomes, and I'll show you several of these in a while. But for the autosomes, all of the rest of our human chromosomes, we are very intolerant of aneuploidies. Here, let's just see if this works. These aneuploidies arise because of mistakes committed during meiosis. So gametes inherit the wrong number of chromosomes, and they may inherit the wrong number of a particular pair of homologous chromosomes. Oops, sorry. See if I can get it to work. So you won't be able to hear it, but I'll show you. Watch the arrow there. Watch this one. There. That never got to the plate. Right. So it's never attached properly. And it just wanders around and it gets included in the new nucleus. Let's see again. These are all segregating properly, but it's hanging around behind. Even that one joins up, and there it is there. Now it's going to be included in the new nucleus. And it's going to give this nucleus the wrong number of chromosomes. Okay, so there's two places in meiosis where this is really important. There's two places where it can occur. The first is in meiosis 1. So in meiosis 1, what you need to remember, at anaphase 1, whole chromosomes separate from one another. There, this is supposed to go that way, this goes that way. But look at this pair here, they never segregated. This here, they failed to move apart. This we call a non-disjunction. And so they both get pulled to the side over here. So this organism here has just got 2 pairs of chromosomes. So there should be 2 chromosomes in the one cell, 2 chromosomes in the other cell. But have a look here, and what we end up with instead is we end up with 3 on one side, 3 in the one cell, and 1 in the other. 1, 2, 3, 4. 1, 2, 3, 4. It's led to an error in the distribution of the chromosomes, and instead we now proceed, this one, we're going to proceed to meiosis 2. We're going to separate these chromatids, make our gametes, and instead of each of the gametes in this example, having 1, 2, 1, 2, we end up instead 3, 3, 1, and 1. So our number is completely wrong. We end up with, instead of being haploid, we end up with being one less than haploid, n minus 1, n minus 1, one more than haploid, n plus 1, n plus 1. These gametes can actually go on to mature. They can actually form a mature egg, or they can form a mature sperm. And when they are then fertilized, they're going to have the wrong number of chromosomes. And this may appear, we'll see in a while. Here's another way it can happen. This was at meiosis 1. We can also have non-disjunction at meiosis 2 there. And in this case, we're supposed to be separating identical chromatids from one another, there and there. But here we're not. We're pulling both of these chromatids across to the one side. And again, we're going to end up here. This is abnormal, 3, n plus 1. This is abnormal, n minus 1. And this one here went on to form new normal cells. Now just imagine if we now get this gamete here and we fertilize that gamete there with it. We're going to end up with 3 pairs of one and 2 of the other, which can actually be, it depends on which chromosome it is, but it can be disastrous. In human beings, these aneuploidies produce very distinctive phenotypes. But unfortunately, in human beings, most of them are fatal, either in the womb or shortly after birth. There are only a few of these aneuploidies in the autosomes, not the sex chromosomes, but in the autosomes, that are tolerated at all in human beings. And the one which we know very, very well, which is tolerated extremely well, is this one, trisomy 21. And trisomy 21 is, there are 3 copies of chromosome number 21, and this produces Down syndrome. Now, you can have other combinations. Trisomy, so Down syndrome is a trisomy, it's a trisomic aberration. 3 copies of a particular chromosome. You can have monosomic as well, where you only have 1 copy instead of a homologous pair. Again, there are a couple which are tolerated, but most of them have very serious effects. There are other things that can happen to chromosomes. The chromosomes themselves can be damaged, especially during cell division. During meiosis, for example, the crossing over process in prophase 1 is sometimes inaccurate and can lead to chromosome damages. We recognize things like deletions, where a chromosome fragment is entirely lost. Sometimes there are duplication errors during replication of the DNA. Sometimes, probably also due to crossing over effects, a set of genes can be actually inverted on a chromosome, and that affects the gene expression. And we also see sometimes bits, instead of, for instance, during crossing over, normally it's done very, very accurately. So the same gene segment, it swaps with its homologue exactly across. Sometimes it goes a little bit wrong, and a gene segment can be shifted on the chromosome or even moved to another chromosome. And this produces some very, very distinctive phenotypes in human beings. So here's the one place where these aneuploidies are very, very well tolerated, and that is in the sex chromosomes. And these non-disjunctions are more likely in the sex chromosomes, especially because the X and the Y, for example, are not completely homologous. They differ from one another. They still should behave as homologs, but they are more liable than other chromosomes for these non-disjunction events. There are some which are very, very well known. One of them is one of the commoner ones. I don't want to say it's common because it's not common in the overall population. But if you're talking about aneuploidies, it's one of the commoner aneuploidies, is Klinefelter syndrome, and that is XXY. 2 X chromosomes and a Y chromosome. It produces a variety of phenotypes, but usually they associate people with Klinefelter being tall and gangly. Klinefelter's is an interesting one because of its societal associations. Somebody once did a study in prisons and thought that they saw more Klinefelters in prison than in the general population, and they came to associate Klinefelters with antisocial behavior. It's completely untrue. It's completely and utterly untrue. Most Klinefelter people don't even know they have Klinefelter syndrome, and they often Klinefelter males, they're always male because they have the Y chromosome, despite the fact that they have 2 X's. They're male because they have the Y chromosome. Most people with Klinefelter discover that they're Klinefelter syndrome when they have problems conceiving. So they go for fertility examinations, and that's when they do the genotype and they find that the husband is Klinefelter. They can still have children, but it may be difficult. There are also yes, go ahead.
Does this disease affect like the health of your sperm or like the fertility of your semen?
Yes, it's the sperm. It affects the actual production of sperm. They have difficulty going through meiosis successfully.
Mm-hmm. And then there's less amount of sperm in the
correct.
Okay.
Yes, yes. There's less sperm that can successfully fertilize. So, but it's not zero. It's not zero. So they can, often with difficulties, they can conceive. I mean, they can have their partner conceive. There are also XYY males, and this does not, again, there's no particular phenotype associated with it, but they again may have problems with fertility because of this introduces difficulties during meiosis. 3 X's have been recorded, female, all female. And this is an interesting one. You remember I told you about monosomy, that is where there's only 1 copy of what should be a homologous pair. It can happen that you end up with 1 X chromosome instead of 2. And this is a very distinctive syndrome called Turner's syndrome. And they are infertile. And there are particular physical characteristics to people with Turner syndrome. There are all sorts of variations on this. There's even people with 4 copies of X chromosomes and 1 Y have been recorded, and still male. As long as you have that Y chromosome, you will still be male. So here's a carrier type for somebody exhibiting Down syndrome. And it's useful for us just to remind ourselves of what the human carrier type looks like. Here we have, these are all the autosomes here. And there are 22 pairs of autosomes. They are identified by the length and structure of the chromosomes. And this is chromosome 21. And you can see that there are 3 copies, 1, 2, 3, where everyone else has 2. Everywhere else there's 2. Here are the sex chromosomes here. This is a carrier type for this little girl here. And so she's XX, so she's female, but trisomy 21 here. And as I say, trisomy 21 is one of the very few trisomies of the autosomes which is tolerated. There are 1 or 2 others. There's a trisomy here, I believe, and I'm not sure, can't remember 2 or 3, both of which are fatal to the fetus. We do not tolerate these trisomies, but trisomy 21 a huge range of phenotypes. They do have a particular look as a result of the trisomy. Mental ability varies tremendously, and there are many, many people with Down syndrome with completely normal intellectual capabilities. But I would say the one thing I've always noticed about Down syndrome, people with Down syndrome, is their innate cheerfulness. They just seem to be such happy people. And I think that's something advantageous. There may well be something advantageous to having people with Down syndrome in our society. Okay. The one thing, Down syndrome, about 1 out of every 830 children are born. Frequency of Down syndrome is associated with age of the mother at conception. But that's never been clearly explained why that is the case. But it is one of the reasons that people may have amniocentesis, to check the chromosome number of the fetus to see whether or not it is Down syndrome. Okay, so hang tight, because I'm just going to switch, if I can find it. No, that's the wrong one. Where am I? Just give me 1 second, will you? I just need to right, let's see. Oh, for goodness sake, where is it? Oh, try this again. Excuse me bumbling around trying to find this. There we are. All right. So we're going to move on. Oh man. We're going to move on to starting to discuss the actual molecular basis of inheritance. We've seen the Mendelian inheritance, everything. We've looked at it in human beings, some of the traits and everything. And now we've also understood how these characters are associated with the chromosomes themselves. And we can see the way the chromosomes behave during meiosis explains the patterns of inheritance that we've noted from Mendel and everybody else. But what we really need to understand is that the chromosomes are vehicles. They are composed of the substance DNA. And the genes that they are carrying are actual genetic sequences. They are sequences of DNA that are actually form particular recipes that need to be communicated to the cell in order for the processes of the cell to continue. We need to start thinking about the DNA itself and how it works and how it functions. So that's going to form the subject of several lectures, but I want to get a good start today. For a very long time, we've known that DNA is a component of the nucleus. That is how it got its name, nucleic acid. There were workers who extracted DNA from the nucleus in the 1800s. They had no idea what it was doing there, but they knew it came from the nucleus and that it was slightly acid. It's a very weak acid. So they called it nucleic acid. But they worked out not structure or anything like that. but they worked out some of its composition, and they realized it's actually quite a simple molecule. It's a very big molecule. Its composition is actually quite simply made. Also present in the nucleus was lots of protein. And the reason for that is we have a huge amount of DNA, these long strands of DNA that have to be carefully organized. And they are organized by winding them around proteins called the histones. And we can then take the histones and start winding them around one another. The function of DNA was completely unknown. They just thought it's too simple a structure, it's too simple a composition to actually do anything really useful inside the cell. It was only with this gentleman here, Frederick Griffith, that we began to get the idea that DNA may, in fact, be doing something very, very important. And Griffith was working on a bacterium called Streptococcus pneumoniae. You'll guess by its name, Streptococcus causes a pneumonia. But there are, in fact, 2 strains of Streptococcus, and only one of them causes pneumonia. Only one is pathogenic. So yeah, go ahead.
So this is just strep, right? Or this is a different type of strep?
No, this is a different strep. Yeah. This is a Streptococcus pneumoniae. It specifically causes pneumonia. It's quite common. It quite commonly does this, but he was interested because he was actually trying to develop a vaccine for Streptococcus pneumoniae. That was how he started working on it. In any case, so he did the following experiment. He recognized that there are 2 strains of Streptococcus. The first strain is the pathogenic one, and it has a thick, sticky capsule. It occurs in, these are 2 little this is 2 coccoid cells together. It's a diplococcus. And they have this capsule around them. The capsule is sticky, outer covering. When you can grow this very easily on a agar plate. And when you grow it, this makes a glistening, smooth colony. So this strain, he called the S strain, or smooth. The S stands for smooth. The other strain does not have a capsule. Here are the 2 cocci, but it has no capsule around it. And when you grow it on agar, it looks dry. It looks rough. So he called this rough R. And only the S, only the smooth can cause an infection. The rough do not cause infection at all. That was one of the reasons he was interested in them. He thought he might be able to use these to make a vaccine because they didn't cause illness. But otherwise, they're identical to the smooth. So here's what he does. First of all, he demonstrates that when you inject smooth into a mouse, the mouse dies. When you inject rough into a mouse, the mouse survives. That's just what you expect. Now he found that he could take a culture of smooth bacteria here. He takes a rich culture of smooth bacteria and he heats them up. In fact, he heated them up, cooled them, heated them up, cooled them, heated them up, cooled them several times until he was sure that they were all dead. He checked that by plating them onto agar, and he could not get any living cell. There were no living cells left. If he took that solution of heated and cooled smooth bacteria and injected them into mice, nothing happened. The bacteria were dead. But now he took it one step further. And what he did was he mixed this mixture, heat-killed bacteria, with this culture here of living rough. He mixed them and he injected them into mice, and bingo, the mouse died. The mouse died. Why did the mouse die? There's no living pathogenic bacteria. The only living bacteria he's injected are these rough, non-pathogenic bacteria, yet the mouse died. And the answer came when he extracted bacteria from the dead mouse, because the bacteria that he extracted were now smooth. They were smooth bacteria. So initially he thought he'd made a mistake. He thought that some of these must have survived. But he then conclusively proved that that is not the case. The smooth bacteria are all dead. They are all DEA dead. They're all dead in this culture. The only living cells are the non-pathogenic. He also thought maybe if he injected enough of the pathogenic, non-pathogenic, still kill the mouse. Wasn't true. He could inject as much rough as he liked. The mice never died for a very good reason, which I'll explain in a minute. Yet these mice injected with a mixture died, and when he extracted bacteria, he extracted living smooth cells. He described this as transformation. He said that these rough cells have been transformed. Somehow or other, he did not know how. Somehow or other, these rough cells have been transformed from rough to smooth. Well, the answer is that these rough bacteria here are acquiring genetic information. When you heat up the smooth cells, the smooth pathogenic cells, and kill them, a lot of them burst and they release all of their DNA into the solution. And when you then mix living rough cells with them, these rough cells are actually able to take up some of that DNA and they incorporate it into their own genome. They acquire the genes for smoothness and they become pathogenic. Now, there's two reasons why this capsule, the smooth capsule, is important. The first is it allows the bacterium to escape the mouse's immune system. The immune system does not react strongly to encapsulated bacteria. That's number one. The second is that this capsule allows the bacteria to stick to surfaces, to stick to mucous membranes. And bacteria very often require adhesion to a surface in order to grow properly. And so this is conferred by a small set of genes which allow this bacterium to be pathogenic because it produces this capsule. Those same genes can be acquired by these rough bacteria, and that transforms them into smooth. This is a critically important experiment in all of biology, but especially in molecular biology. This was the first demonstration we ever had that many things are going on, but first of all, that these pathogenic characters may depend on genes which bacteria can acquire from other bacteria. You can actually end up passing these genes around a bacterial population and transforming them into pathogens.
Professor?
Yes, go ahead.
At the end, I mean, we don't have a streptococcus vaccine, right?
Yes, there is.
There is a streptococcus vaccine?
Yes.
For
yes.
for
streptococcus pneumonia, yes. He actually did end up producing one himself, but it wasn't a very good one. Mm-hmm. But there is now a modern one, which is very, very good, which you can have. Probably at your age, I don't know, you could ask a doctor, but I think it's normally only recommended. I just had mine in the last couple of months. They recommend it as you start to get on in years. They recommend that you have you get your.
I know all the vaccines that I've gotten, and I never saw that one on the list. I know that.
No, you won't. No,
right.
no. It's. I think it's mostly recommended as you get older.
I see. And then because usually I know when I was younger, I used to get strep throat all the time because kids were, it's very contagious and kids used to spread it around at school. I remember if there was strep, then all the kids would get strep. You usually just, since it's a bacterial infection, you could usually cure it with just antibiotics.
Correct.
It's not a virus.
That's another strep. It's usually one called Streptococcus pyogenes that causes strep throat. There are a couple of different streps, but this Streptococcus pneumoniae is specific for this pneumonia. often as a secondary infection after influenza or something like that. In young people, you may briefly have it and clear it without really knowing that you've had it. But in older adults, it tends to accumulate in the lungs and then spread through the lungs as a proper pneumonia. It can, in very old people, it's extremely dangerous. It's extremely dangerous. Okay, so just to point out to you, that he knew nothing about DNA. He didn't, for many, many years, make the association between what was happening and DNA. It was up to other workers to actually redo his experiments. Nobody believed his experiments, by the way, until they repeated them and everybody got the same results, which is the way science should work. But other workers were fascinated by this, and they found there was a big argument because the nucleus had lots of DNA, but it also had lots of protein in it. There was some argument about whether the hereditary principle, the genes that were conferring this information, were composed of DNA, nucleic acids, or whether they were composed of protein. The protein camp was much more popular. Most people thought that heredity was conferred by protein in inheritance of protein itself. They thought DNA was too simple to convey all that information. But somebody redid this experiment, and what they did was they took this mixture here, before they injected it into mice, they treated one lot with an enzyme which cut up DNA and destroyed it. And then they took the other solution and they treated it with an enzyme which destroyed enzymes. And the one where they destroyed the DNA, the mice survived. The one where they destroyed the proteins, the mice died because this was still being transformed. The DNA was still active. So that was a conclusive demonstration. It was DNA. It's DNA that gives this information. What is happening here is that these bacteria here are inheriting genes from these dead bacteria. And those genes are composed of DNA. And the recipes for all the metabolic processes that will make this capsule are in that segment of DNA that these have inherited. Okay, so I think I'm going to stop there. And I'm going to proceed as far as I can with this and with the next section as well on Wednesday. I do need to do a little bit of catch up because we've fallen behind a bit. But your test on Monday will examine up till Wednesday. Okay, it will examine the stuff we do on Wednesday. And then we'll continue, of course, thereafter. I may, at some point, if there's not strong objections, I may put in an extra class. I will record an extra class just to help us catch up. Okay, so let's leave it there, and then I'll see you on Wednesday. Unless, of course, there are questions, you're more than welcome to pose any questions.
Professor?
Yeah.
So I had a question. We're having a quiz, right, this Wednesday?
Yes, you will have a short quiz this Wednesday.
Okay. I asked you last week, but I just wanted to make sure to remind you, would you be able to make, when we take the quiz, so it would show us the answers?
Didn't I put them all up?
No, I mean, like, when we take the quiz, it usually at the end it says like the answer, so you could see what you got wrong.
Oh, okay. Yes, I think I can do that on Canvas. Yes.
Great, thank
you.
Yes,
but I did post answer sheets.
No, I saw the answer sheets, but I couldn't really understand what I got wrong. You know what I mean? So I couldn't really understand what I got wrong, what mistake I made, and how can I change the answer.
All right, then. I'll see that I can do that. Yeah.
Great,
okay.
Thank you.
Thank you, Professor.
Thanks. See you all again.
Thank you.
See you on Wednesday.
Have a good day.
Bye-bye.